Chemical processes within a neuron are managed by an architecture in which the higher levels determine the availability of chemicals for the more detailed levels. You can identify 4 clear levels to this architecture: the chemistry controlling the deployment of genetic DNA information in the nucleus of the cell, the activities of messenger RNA that carries segments of this DNA information throughout the neuron to control construction of proteins, the management of the construction of the proteins, and these proteins control the major functions of the neuron in various ways, and a wide range of chemicals that implement these functions that are synthesized by under the control of the proteins. The primary source of information for a neuron, as for any other cell, is the DNA genetic information that's stored in the nucleus. A large and important part of this information is code that allows construction of different proteins. The information in the DNA is accessed by activating a segment that codes for one protein to drive the synthesis of messenger RNA. The messenger RNA can be used to synthesize the protein, and that synthesis can occur in the vicinity of the nucleus, or the mRNA can be transported to distant locations of the neuron, like an individual synapse, and used to synthesize protein for use only in that location. The proteins that are synthesized play a wide range of different roles in the neuron. Proteins like actin form the skeleton of the neuron, giving it shape and driving changes like axon growth or expansion of synapses. Functional units like G protein receptors, G proteins, and ion channels are also proteins. And proteins called enzymes catalyze all the chemical reactions in the neuron. And one important class of enzymes is kinases that activate other enzymes. One example, called protein kinase C, is illustrated. A kinase activates another enzyme, and that other enzyme could itself be a kinase that activates yet other enzymes, allowing complex networks of control over cell chemistry. Enzymes also catalyze the synthesis of other chemicals, and also the reactions between those chemicals, exerting detailed control over the neuron. One class of chemicals synthesized under enzyme control is the neurotransmitters like glutamate or GABA. Another class is second messengers like AMP. Proteins like kinases control other proteins, and proteins regulate the processes by which proteins are synthesized from mRNA, messenger RNA, and even the processes by which mRNA is synthesized from DNA. So there are feedback loops within this chemical architecture. At the detailed level, a chemical process that creates some chemical from whatever's already available in the environment will generally only take place if an enzyme that catalyzes the process is present and activated. So the chemical glutamine is widely available, but glutamate will only be synthesized from glutamine if the enzyme glutaminase is active. If the enzyme glutamate decarboxylase is present, GABA neurotransmitter will be synthesized from glutamine. Another universally available chemical is ATP. It's the source of energy to drive chemical processes. If the enzyme adenylyl cyclase is present and activated by an active G protein, it will convert some ATP molecules into the second messenger, CAMP. Proteins, including enzymes, are long chains of amino acids folded in a complex but very specific way. enzymes catalyze a specific chemical reaction, and within the folding there's a site or sites at which the enzyme exerts its catalytic action, for example by binding to the chemicals that participate in the reaction. Often, activation of this binding site requires attachment of a phosphate group at some other specific location, and removal of the group deactivates the enzyme. Although it's true that for a few enzymes the opposite is true. Removal of the phosphate activates them. As mentioned earlier, a kinase catalyzes the addition of the phosphate group to a specific other enzyme. And there are also phosphatase enzymes that catalyze removal of the phosphate group from specific enzymes. DNA is made up of two strands of sugar chemicals wound around each other, and one stretch of DNA corresponds with one protein. The mRNA needed to create the protein is synthesized by a transcription complex that moves along one strand of the DNA, synthesizing the corresponding messenger RNA. And the transcription process is triggered by chemicals that activate the complex. These chemical factors are themselves activated by enzymes. After activation, the transcription complex moves along the DNA strand, generating the messenger RNA. Once the mRNA is complete, it breaks away from its DNA template. The messenger RNA is transported to a region where the chemicals for protein construction are available. The mRNA must be activated by a chemical factor, which is itself activated by an enzyme. And of course, the enzyme will need to be activated, and so on. Once the mRNA is activated, the corresponding strand of protein is synthesized. Following synthesis, it breaks away from the messenger RNA template, and when it breaks away, it folds into its complex structure without further guidance. The folded form is implicit in its linear structure. Now, an external neurotransmitter can trigger a cascade of chemical reactions that lead to activation of DNA to create new proteins. For example, a G protein receptor can act via second messengers to activate a kinase. That kinase can activate another kinase, and so on, down to a kinase that can act on activation factors to trigger transcription of messenger RNA.